Good morning, everyone. We are from Universiti, Malaya. Today, we'll explore Naringin, a natural compound found in citrus fruits with strong antibacterial properties. According to Shaver, 2020, it can disrupt bacterial cells and inhibit crucial enzymes, making it a promising antibacterial agent. Antibiotic resistance is a growing problem in clinical centers throughout the world. Incorrect and unnecessary usage of antibiotics increase the risk of antibiotic resistance in different societies. What polyphenol actually is? Polyphenols, natural compounds in plants, are potential alternatives to conventional antibiotics for combating antimicrobial resistance. They exhibit antimicrobial properties against bacteria, fungi, and viruses. Naringin found in citrus fruits has shown remarkable antibacterial properties. Quercetin from medical plants like ginkgo, red onion, and kale can be used to reduce the activities of enzymes and toxins and interacts with the cytoplasmic membrane or lowers pH values. We have two research questions. Firstly, is the antibacterial potency of quercetin and naringin increased when these flavonoids are used together compared to their individual antibacterial effects against E. coli? Secondly, can the combination of quercetin and naringin offer a novel antimicrobial strategy to address infections caused by priority pathogens like E. coli? We have two main objectives. Compare the antibacterial effects of combined quercetin and naringin against E. coli versus their individual effects. Determine if this combination enhances antibacterial potency. Our methods include broth microdilution assay to find the minimum inhibitory concentration, MIC, and minimum bactericidal concentration, MBC. Besides that, agar well diffusion to visually observe antibacterial activity. In the lab, we used agar well diffusion to see how our compounds inhibit bacterial growth. We prepared plates, added bacteria, bored wells, and added our samples. After 24 hours at 37 degrees Celsius, we measured the inhibition zones. Our results showed that naringin and quercetin, especially when combined, effectively inhibited E. coli. Larger inhibition zones were seen with higher concentrations, indicating a stronger antibacterial effect. At low concentrations, 0.1 moles, neither naringin nor quercetin, nor their combination, inhibited bacterial growth. At 0.5 moles, quercetin alone created a 9 mm inhibition zone, while the combination of naringin and quercetin produced a larger 15 mm zone. At 1 mole, naringin alone showed a 10 mm zone, quercetin had a 14 mm zone, and their combination resulted in the largest zone of 17 mm. These results suggest that combining naringin and quercetin significantly enhances their antibacterial effect, especially at higher concentrations. We used the broth microdilution assay to determine the MAC and MBC of naringin and quercetin. We diluted the compounds, added bacteria, and measured turbidity after incubation. Lower turbidity means better bacterial inhibition. In this test, we used a 96-well plate with different concentrations of naringin, N, quercetin, Q, and their combination, N plus Q. We started with a high concentration and diluted it down to a very low concentration. The lowest concentration with no visible growth is called the minimum inhibitory concentration, MIC. This tells us the smallest amount needed to stop the bacteria. Here are our MIC results. The MIC, or minimum inhibitory concentration, is the lowest concentration of a compound that prevents bacterial growth. This result shows that the combination of naringin and quercetin is more effective at lower concentrations than naringin alone, indicating a potential synergistic effect. The result analysis of the study. The result analysis of the summary of well diffusion findings. 1. The combination of naringin and quercetin showed the largest inhibition zone, indicating the greatest antibacterial effect. 2nd, naringin alone was the least effective. 3. Antibacterial activity increased with higher concentrations of polyphenols, showing larger inhibition zones at 1 million. Comparison with previous studies. 1. Antibacterial activity test by agar well show concentration-dependent inhibition increases in inhibition zones. 2. Quercetin showed moderate inhibition in previous studies, while naringin had minimal effect against E. coli. This matches our findings where quercetin was more effective than naringin alone. Here is the implications keys of our findings on naringin and quercetin antimicrobial potential. Firstly, the novelty of our study is the combination effectiveness, which naringin and quercetin together show significant bacteriostatic effects against E. coli, acting as a dynamic duo. Besides that, comparative potency where the MIC of naringin combined with quercetin rivals that of quercetin combined with meropenem, a known antibiotic. Secondly, benefits of intended users and society is using naringin and quercetin as natural antibacterial agents offers sustainable and eco-friendly alternatives to chemical antibiotics, benefiting agriculture and public health. This combination can lead to effective treatments with fewer side effects, improving public health and reducing healthcare costs associated with antimicrobial resistance. Here is our commercialization potential of our study. These promising results suggest a market opportunity for developing new antibacterial products. Naringin and quercetin could reduce dependency on traditional antibiotics and combat antimicrobial resistance. For future research, while quercetin and naringin show potential to enhance antibiotic effectiveness, more research is needed to determine their impact on other bacterial species. In conclusion, our study suggests that naringin and quercetin could lead to new effective antibacterial treatments with substantial market potential. Thank you.